Hi, everyone, and welcome to our podcast. My name is Laura Vidal. I'm VP of Oncology at Fortrea, and today we want to talk about the evolving role of imaging in precision oncology. And to do so, we have invited Dr. Ron Korn, founder and chief medical officer of Imaging Endpoints. Thank you, Ron, to be here today. Maybe before we start, you can give us a bit intro about you and your company.
Well, great. Thank you, Laura. And it's a real pleasure to be here. Imaging Endpoints is a full-service imaging CRO that was founded about 16 years ago, really to make a difference, to bring the best of radiology sciences to drug device development. By background, I'm a nuclear medicine physician and radiologist, and prior to that, I have a PhD in cellular biology. So for an imaging core lab, integrating the best of science with radiology is what we do best at Imaging Endpoints.
Thank you. Perfect. So both of us work very close on oncology, and in fact, we met at ESMO in Berlin last month. So
Yep.
For some of the presentations that we have seen and the clinical studies, we recognize that imaging is becoming a dynamic tool, guiding drug development and being more relevant, particularly in designing clinical studies. So based on your experience, can maybe share some of the latest advancements in imaging that are helping to better refine patient selection for clinical trials and as well improve ability to identify those that are early responders?
That's a fantastic question and one that we're very excited about. Advances in molecular imaging are truly transforming how we identify and stratify these patients for clinical trials. These innovations are really allowing us to have more precise selection of the patients, ensuring, as the statement goes, the right patients are enrolled at the right time. And those assessments really begin to bring the treatment journey in better focus. A clear example is PSMA PET imaging for prostate cancer. Now, Imaging Endpoints played an important role in helping define the use of PSMA PET in clinical trials to identify patients who may be candidates for this type of new therapy, the radioligand or radioisotope therapy. And that approach of using PSMA PET really proved essential during the clinical development of these agents. And that has then led to being translated directly into clinical practice, improving both patient outcomes and therapeutic targeting. Similarly, imaging can really be leveraged to identify early responders. And we have been deeply involved in this for over a decade, beginning with the use of FDG PET in pancreas cancer when we were working with the IMPACT trial to show that an early cooling of FDG PET in metastatic pancreas cancer really portended for a good outcome with that drug. And now that's expanded to new modalities, such as MRI-based technologies using diffusion-weighted imaging or dynamic contrast-enhanced MRI to look at perfusion, along with a lot of PET kinetics that can be evaluated, can be used to evaluate changes in the tumor and their microenvironment. And I think one of the interesting things that is being developed for the tumor microenvironment has come out of the EANM, in which the targeting of fibroblast-associated proteins, which produces this fibrotic environment for tumors from the cancer-associated fibroblasts, is becoming a real target for drug development and therapy.
I'm going to stop you here because I'm feeling that the people that is listening to that podcast, maybe they are wondering, and what happened with CT scan and RECIST? Because RECIST has been the cornerstone for assessing drug response for forever. But we all know that it has its limitations. So maybe I would love to hear your thoughts about this new technology like artificial intelligence, radiomics, that everyone is talking about on how to interpret much more complex imaging data sets and how can that improve our RECIST system.
Great. Well, it's a great question, a very important and relevant question. Well, RECIST and other established criteria such as RANO criteria for brain or Lugano for hematologic malignancies, they'll continue to serve as the primary frameworks for assessing therapeutic response. But these newer strategies, particularly volumetric assessment that's really focused on things like tumor growth kinetics or quantitative imaging biomarkers such as radiomics, often can detect responses earlier and better, and they also reflect the overall disease burden and their changes. Now, artificial intelligence is really playing what I think is a very meaningful role in drug discovery, from identifying novel targets to optimizing compound development. But in imaging, AI offers the ability to integrate multiple complex and often different data sources, such as imaging metrics, laboratory and clinical outcomes, et cetera, into a unified assessment for both safety and efficacy. And there have been attempts, I think people always ask us about artificial intelligence for resist. Well, there have been attempts at auto resist with AI, which is still in the early stages and showing promise with lesion detection measurements, but they're only being successful currently in the highly controlled and limited settings. So it does hold promise, but there's a long way to go, especially for pivotal trials. I think at ESMO was a really great meeting, ESMO 2025 in Berlin, because there was a lot of presentations which really showed the promise of AI, especially in integrating imaging with molecular and clinical data. And there are a couple trials that I think stood out in my mind as being noteworthy. The first was the NERO trial from the UK in mesothelioma, which is, as you know, is a notoriously hard tumor to measure by RECIST. And the folks from the UK showed that when you integrate tumor volumes along with intertumoral heterogeneity, it really helped identify the patients that were most likely to respond to niraparib versus active symptom control. You can then look at the Aegean trial from the US, and John Haymark looked at changes in the radiomic features in resectable lung cancers, particularly what's called the delta energy of the primary tumor, which is sort of a measure of heterogeneity, at screening to surgery to predict complete pathologic response. And when adding ctDNA to that equation, it really improved the prediction better. So in other words, AI is stratifying patients by risk and response likelihood better than, I think, standard imaging metrics.
That sounds super interesting. And maybe as well on the same line, and based on your cancer biology expertise, we are seeing that non-invasive methods are increasingly used to predict early response together with imaging. So on those trials where both imaging and liquid biopsy are used to monitor patient response, how do you ensure that those two different types of data align? And what are the challenges that we are facing when we're trying to integrate and interpret results from those complementary modalities? What's your view on that?
Right. So the key word here is coordination. You really have to ensure that there's alignment between the imaging and the liquid biopsies. And that really begins at the protocol development stage. It requires early collaboration across the scientific, clinical, and operations team to make sure that the study endpoints are well defined, clearly delineated, and appropriately customized to demonstrate the efficacy that you're looking for. At IE, we build in this alignment by making sure that our imaging review charters, both its design and setup, align with the protocol. The imaging manuals, including the schedule of events, are in sync with the different biopsies or different tests that will be performed, that the ECRF design is appropriately configured so that it's easy to integrate both the imaging and the liquid biopsy data, and as well as making sure that there's training on all aspects. Establishing these foundations really helps, in my opinion, to ensure that the imaging and molecular data are being collected and interpreted in a synchronized fashion. And what's really important, and I can give you an example, is that when you plan to obtain the liquid biopsy results along with the imaging, it's really important that you look at the logistics of when each of those different tests are being performed. As an example, in one clinical trial, it was very important to acquire the liquid biopsy on a day that was after a very long imaging session. Well, that adds a lot of undue burden to the patient, and it really causes a reduction in enrollment. So working with the team, we made sure that the imaging and the liquid biopsy was really obtained in a temporal manner to ensure patient compliance with those requirements.
Thanks, Ron. I think that that really makes sense, and probably it's going to probably lead what we will see more and more into the upcoming years. Let's change a little bit and talk about the rapidly growing field of theranostics, that approach that combines therapy and diagnostics using the same radioligands to find and treat cancer, as you mentioned and put an example for PSMA and prostate. This approach comes with several hurdles from different angles: regulatory, safety handling, other radioactive materials, dosimetry. Can you provide your view and explain how companies like you are gearing up to manage these challenges as theranostics become more mainstream in clinical trials?
Fantastic question. This is really essential for the success of theranostic trials in general. And the way that we approach this is using a structured yet flexible approach that combines certain standardization with adaptability. So first, what we do is we are focused on establishing a well-defined, standardized framework for implementing the theranostic trials. We want to ensure that they're consistent, that there's high quality, that all staff members from both the imaging CRO, the clinical CRO, and the site are adequately trained, and that there's compliance to the protocol across all the sites. At the same time, we recognize that every study has unique scientific and logistical needs. So we need to provide customization when necessary to accommodate these protocol-specific requirements. As a perfect example, in many of the first-in-human studies with the theranostics, it's very important that you collect good data on the biodistribution and dosimetry of the product that you're injecting. And that often requires multiple time point imaging, usually more than three or more time points. As data evolves, you can then adapt your clinical trial to reduce the number of imaging time points so that you could still get the same type of data, but also make it easier on the site, on the patient, without compromising quality. Another example is that we really look hard and carefully at the preclinical data that's brought forward to us to make sure that when we translate preclinical data to the clinical environment, that we don't see anything that's unexpected or untoward that we did not see in the preclinical data. So looking at the early biodistribution is critical, and one of the challenges that a lot of folks have when performing these theranostic studies. So in summary, I guess I would say that really making sure that you have a structured yet agile model for conducting these trials really enables us to manage the growing complexity of these theranostic studies while ensuring that that we have the quality, efficiency, and patient safety as primary focus in every trial we do.
Thanks. And finally, I think that you have already explained a lot of the innovation that we are seeing, but in your opinion and looking ahead, what are the most exciting or promising trends in medical imaging that you will believe transforming precision oncology over the next 5-10 years? And I'm a visionary, so I'm expecting like a visionary answer from you. So let's see.
Well, this is something that you and I could discuss for hours on end because it's really an exciting field right now. And looking ahead, I no doubt molecular imaging is going to continue to shape the future of precision oncology, not only in cancer, but across many disease areas. And it's really its ability to visualize the biologic processes in real time that remains central to how we're going to diagnose, treat, and monitor patients. At the same time, AI is really playing an increasing transformative role. And I look at it in 3 or 4 areas. First, I think AI is really impacting the acquisition of imaging, enhancing image quality and harmonization of data across scanners and sites. And that's really important in a multi-center trial, for example. Really interesting that I keep an eye on is what I call AI transformation, which is enabling cross-modality convergence. For example, some investigators are now generating MRI data from CT scans and vice versa. Getting an MRI scan can be transformed into CT data. So this really expands diagnostic flexibility. I think we've already touched upon the interpretation of how AI can support our radiologists and nuclear medicine physicians through their advanced automated platforms to enhance rather than replace. And that's the key word, replace expert review. And I think the final area that I'm really excited about is the practical development of all this new technology. AI is really bringing together real world imaging data and what is called digital twin concept, which actually came from NASA. And a digital twin idea combines imaging and other omics to model a patient's disease and test whether a particular therapy is likely to succeed. So this could really radically define eligibility and efficacy signals before the trial even starts. And I think the real awareness of all this technology has really brought a lot of attention to our regulatory evolution. And as you know, in 2022, the FDA came out with a position paper on the new approaches, methodologies, or NAM. And here the FDA wants to accelerate drug development even, and I think eventually eliminate animal testing by leveraging AI-based computational models, human organoid model-based laboratory tests, and real-world human data. This is really interesting because it's now being implemented for monoclonal antibodies where accurate AI models and alternative preclinical models can predict how a monoclonal antibody will react when injected into a patient. This is where imaging is going to play a very important role because I think there's a real opportunity for the molecular imaging industry to make these new agents for looking at the biodistribution outside of these preclinical models, because you still have to perform first-in-human studies, and having a SPET where PET scan is well suited to confirm the biodistribution, looking for any off-target or potential safety concerns. So ultimately, I would say the goal here in the future is to cure disease and, more importantly, to restore health. And imaging is going to remain a vital bridge in that journey. So over 15 years of being at Imaging Endpoints, I've been proud to contribute to this progress as our company really shows our passion of connecting imaging to the cure, which continues to drive everything we do.
Wow, that was quite such a statement. So thank you, Ron, for sharing your insights with us today. It's truly exciting to see what's ahead. I think that pushing innovation in imaging is pushing as well precision oncology, and maybe, as you are seeing, even move to non-oncology aspects. So thank you again, and to our listeners, thank you as well for joining us today.